Since the 1920s, the Darwinian idea has been reduced to gene selectionism. The mainstay of Darwin's original thinking, namely adaptationism, has been diminished as a result. And this has doomed modern Darwinism to incoherency. Adaptation is central to any rational conception of fitness, and any theory of evolution that ignores it is doomed to incoherency. A mainstay of modern Darwinism's defense is that no matter how incoherent it might seem, it's backed by solid scientific evidence. Take, for example, the central dogma of molecular biology, which asserts that information in DNA sequence code can flow only one way, from DNA to RNA to protein, and information can never flow the other way. This suits the gene selection idea very nicely. It's only DNA sequence code that can act as hereditary memory and replicate to be passed on to future generations. And it's from translated DNA code into protein that function arises, and with it, fitness. And what is fitness? Well, in a gene selectionist world, fitness streams from fecundity. In Darwin's own conception, fitness streams from adaptation. These are two radically different conceptions of fitness, so radical that there is presently a bright line drawn between organisms and genes, and the Weismann barrier is one expression of this. This leads us to what I've called the central dogma of modern Darwinism, and we can express this with a metaphor of the relationship between chicken and egg. In the modern Darwinist idea, the supremacy of the gene means that the chicken is just an egg's way of making another egg. Because genes are supreme, the actual chicken is irrelevant to evolution. It's just the means to the end of producing a new egg. In other words, the chicken is a mere vehicle for transmitting genes from one generation to the next. For much of the 20th century, this peculiar viewpoint was bolstered by molecular biology and the emerging conception of the gene as sequence code in DNA. This is all beginning to unravel, and as a result, a new landscape is opening for how we think about evolution, non-Darwinian ways to think about evolution, specifically. The more we've learned about the connection between sequence code in DNA and function, the less realistic gene determinism seems to be. Along with that, neo-Darwinism has been getting less realistic as well. We can't go into all the nuances here. This is a vast subject. For one thing, we're interested in evolution here, not molecular biology. Before we can get to evolution, though, we need to sketch out these ideas. For example, a stretch of DNA not only includes sequence code, it also includes punctuation, specifically indicators where transcription into an RNA molecule can begin and the point at which it stops. A messenger RNA transcript is the result of this. The RNA molecule can also be edited in the cytoplasm. Particular segments of RNA, indicated here in yellow, can be cut out, with the remaining ends stitched together into a new messenger RNA molecule. Syntax can also come into play. Just like the meaning of a sentence depends upon the arrangement of words, so too can the segments of RNA be stitched together in different ways to yield entirely different proteins. Finally, there's context. The most famous example of this involves segments of DNA, indicated here in red, that can migrate from one address in a chromosome to another, or even to entirely different chromosomes. There are many other examples of contextual interpretation of nucleotide sequence code, but the end result? The same segment of DNA sequence code can encode multiple functions. This complexity has built up to the point that we have to view the central dogma in a different way. The flow of information from DNA to RNA to protein can still stand, and function can still arise from protein, but we know this cannot be the end of the story. Protein, for example, can feed back onto DNA, closing the loop of information flow implied by those one-way arrows of the central dogma. Replication and all the other functions of DNA, the genome in a word, depend upon various kinds of proteins to manage them. We also know that there are many ways the RNA, or transcriptome, can feed back onto DNA to change how certain genes are transcribed or not. And we know that the proteins of the cell, the proteome, can similarly mediate function of RNA. And finally, there's a sort of self-referential function of each of these complements. Within the genome, DNA can mediate function of other DNA molecules, as in the example of transposons. Within the transcriptome, interactions between RNA molecules are complex, and these can mediate function as well. And finally, different components of the proteome mediate the function of other proteins, even to the point that proteins can replicate on their own, a property that supposedly is limited to DNA. So the overall picture of the interaction between DNA, RNA, and protein is very complex and fluid. In other words, the emerging picture of how hereditary information is translated into function is process-based, not object-based. We've spoken elsewhere about the distinction between object gene and process gene, and it's process gene that is emerging as the scientifically better way to think about it. And with it has come the demise of the idea of gene determinism. DNA can no longer be thought of as a specifier of function. Rather, it is a participant in the complex set of processes that make up cell function, including hereditary memory. This dynamic interplay between memory, interpretation, and function is what underlies adaptation in the individual organism. But individual organisms don't evolve. What we really want to know is what this says about evolutionary adaptation. We might ask, for example, is the only hereditary transmission of information through DNA, say in a segregated germline, as August Weismann thought? Or could the memory embodied in one generation's transcriptome and proteome be transmitted as well to the next generation? Well, patently, it could. The bridge between generations is gametes, such as the ovum. The ovum is a fully functioning and constituted cell with not only a genome, but a highly complex transcriptome and a proteome. What does this have to say about evolution? Well, we have to return to embryonic development and the Weismann barrier. The nuances I've just outlined are so-called epigenetic mechanisms. Epigenetics simply means above the gene, that is, those things that go beyond DNA's sequence code. There's another suite of epigenetic mechanisms that have a more direct bearing on how we think about hereditary memory, adaptation, and evolution. Geneticists have long had to confront a problem. There's much more code in DNA compared to genes that are actually expressed. This means that there must be large stretches of the DNA molecule that are not transcribed into proteins. What determines which parts of the DNA molecule are transcribed and which are not? Let's illustrate with a short stretch of chromosome. What determines whether a stretch of DNA is transcribed or not is a suite of so-called epigenetic markers. I don't want to get too much into the biochemical details here. I just want to make the point that there are three major types of these epigenetic markers. There's DNA methylation, which involves attaching methyl groups to the nucleotide bases. There's also a group of proteins known collectively as histones that wrap DNA and mediate its replication. And finally, the DNA molecule is coiled at places into small knots, if you will, known as nucleosomes. All of these prevent transcription of genes. Epigenetic markers will paint the DNA molecule at various places and prevent transcription there. Only unpainted stretches of the DNA molecule will be open to transcription and translation into proteins. What's germane to adaptation is that these epigenetic mechanisms are responsive to environmental conditions. Depending upon environmental conditions, transcription of different patches of DNA may be suppressed or opened up for transcription. Painting of DNA with epigenetic markers is one aspect of managing function in cells, and it is one way adaptation can mediate cell function. Diet or stress or other types of environmental modifiers can change the pattern of epigenetic markers, producing a persistent alteration of function and adaptation. What we're really interested in here is how these nuances might play some role in evolution. That means we have to confront the same problem August Weismann did, namely, what controls differentiation of tissues during embryonic development. Why? Well, because embryonic development is itself a kind of evolution. We start, as always, with the zygote, and we'll follow the fate of one chromosome. During embryonic development, cell division piles upon cell division, producing the embryo. As the embryo develops into the organism, these lineal descendants of the zygote become first stem cells, which then mature into differentiated tissues. And we'll follow four: muscle, skin, brain, and bone. There are many more tissue types in this, of course, numbering somewhere in the 20s, but let's just keep it simple. The different tissue types are different in structure and function because they have different patterns of expression of their genes. Yet, the same chromosome exists in the cells of each type of tissue. Each cell, no matter what the tissue type, therefore carries the same DNA molecule as every other cell. How to explain the different patterns of expression then? Well, here's where epigenetic markers come in. The chromosomes of the different tissue types have been painted with different patterns of epigenetic markers. In this way, the pattern of silenced DNA differs from tissue type to tissue type. The interesting thing about the epigenetic markers is that the patterns are heritable. That is, the pattern is passed from generation to generation within each differentiated cell lineage. The differentiation is also generally irreversible. This, by the way, is where August Weismann went wrong. He thought silenced genes were not silenced, but destroyed. Which raises the question about those cells in the germline. Because the pattern of epigenetic markers is generally irreversible, this would seem to require that the chromosomes in the germline lineages not have any epigenetic markers, that these be clean chromosomes, in other words. There's an obvious question we need to ask, though. How do the chromosomes in the different tissues come to have their particular patterns of epigenetic markers? We can understand this better if we bring a different perspective to embryonic development. Here's our tree of lineal descent from the zygote to form our four different tissues. Let's remember the concept of the adaptive boundary and the extended organism. Embryonic development is not simply the descent and differentiation of cell lineages. Embryonic development is creating the new environment of the organism, delimited by the organism's outermost adaptive boundary. As differentiation proceeds, the different tissues form their own environments, nested within the larger environment of the organism. Let's take a closer look at one of these tissue types, say the skin. The skin, being a distinctive tissue within the organism, is bordered by its own subsidiary adaptive boundary, which creates a distinctive environment for skin cells and their associated chromosomes. Because the chromosomes of skin cells are embedded within their own managed environments, the accumulation of epigenetic markers there is an adaptive response to this newly created environment. We could put it this way: the burden of epigenetic markers is an acquired characteristic of the lineage of skin cells. Because epigenetic markers replicate with the chromosome, these acquired characters are heritable. This is sounding a bit, dare I say, Lamarckian, to which we have to say, well, so what? We have to follow evidence and logic. If we look upon embryonic development in terms of the extended organism idea, it's actually quite a simple jump to a credible theory of Lamarckism. Remember, the organism is simply one level of hierarchy, separated from the external environment by an adaptive boundary. By the same token, each tissue type provides an additional layer of hierarchy of tissue-specific environments divided from the internal environment of the organism by its own adaptive boundary. If differentiation can be made heritable by epigenetic markers, what then is the barrier to the same process operating at the higher level hierarchy of the organism-environment interface? And if these epigenetic markers are heritable, you've arrived at a fully coherent theory of Lamarckian evolution. Organismal adaptation is now wedded to evolutionary adaptation, not divided from it, as the Darwinian idea would demand. Okay, that's interesting. What's the evidence? An interesting example of heritable epigenetic variation can be found in the evolution of blindness in cavefish, like this Mexican tetra. These fish lack eyes, obviously, but they are close evolutionary relatives to the other tetras, which is a diverse group with hundreds of species, nearly all with fully functional eyes. How does such a drastic evolutionary change, like loss of eyes, arise within such a closely knit group? One could imagine a mutationist explanation for this, some mutation that causes the loss of the eyes, for example, but it's harder to imagine a Darwinian explanation, that is, a slow, gradual, and imperceptible progression generation to generation from eyes to absence of eyes. Alternatively, we can invoke Lamarck's classic concept of use and disuse. Cave animals don't use their eyes, so they degenerate. Lamarck's core idea, remember, was that use and disuse not only occurred within organisms, they were heritable. This is what modern Darwinism rejects, even though use and disuse was central to Darwin's own concept of adaptation. But it turns out that use and disuse can be inherited, and heritable epigenetic markers are the mechanism. How does this work? Well, vision in fish is quite an expensive proposition. Vision costs energy. For fish with eyes, vision accounts for about 1/6 of the fish's total energy cost. If the fish lives in a visual environment and vision helps that fish find food, then there's no problem. Vision helps the fish find sufficient food to pay the cost, so to speak. Caves, in contrast, are not only dark, but typically are very low productivity environments. A fish with eyes living in a cave would therefore be spending 1/6 of its energy budget on a visual system that is of absolutely no value to the fish, and eliminating the eyes would save that cost. So the explanation for eye loss may very well be Lamarckian. Development of eyes during embryogenesis typically involves triggering a developmental pathway for eyes, which I'm showing here as a yellow stretch on a chromosome. Starvation is one of the things that triggers painting of chromosomes with epigenetic markers. Starvation is likely in a fish inhabiting a depauperate environment and with a very expensive visual system to maintain. With each generation, then, the eye development pathway becomes painted more and more heavily with epigenetic markers. These epigenetic markers are heritable across generations, so they accumulate with each generation. The result is an evolutionary emergence of a lineage of fish without eyes. And the blind lineage emerges comparatively quickly because the loss of eyes is due to the accumulation of heritable epigenetic markers from generation to generation. And this can happen faster than mutations in the fish's eye development genes. This explicitly Lamarckian model also opens the door for us to revisit the whole concept of the Weismann barrier. Weismann focused on a model for the germline that we might call the segregated germline. The zygote gives rise to two major lines of descendants: the somatic tissues, which develop into the tissues of the adult body, and the germinal tissues, which ultimately produce the gametes. In Weismann's own conception, the two were segregated, as if there was a barrier between inheritance in the somatic tissues and the germinal tissues. Not all organisms have segregated germlines, however. Plants are an obvious exception. Their germinal tissues arise from what we might call the differentiated germline. Here, again, we have a zygote, which gives rise to the somatic tissues of the plant. The somatic tissues can continue to grow as vegetative growth. This, by the way, embodies its own form of fitness. From the somatic tissues, there occasionally arise specialized germinal tissues, which ultimately give rise to the gametes. Because the germinal tissues arise directly from the somatic tissues, there can be no strict germline segregation. Well, how do Weismann's ideas stack up here? Let's illustrate how. A good example of a segregated germline is the ovarian follicle, the structure in the ovary which ultimately gives rise to the ovum. The ovary itself is a compound organ. It consists of the tissues of the ovary itself and, of course, the cells that will give rise to the ovum. The ovarian tissues are somatic, while the ovum is part of the germline. The ovum doesn't just sit there in splendid isolation, it's enclosed within the ovarian follicle. The follicle consists of cells from the somatic line. Is the follicle then the physical expression of a Weismann barrier, the structure that keeps the ovum isolated from the soma? We shall see. A good example of a differentiated germline may be found in plants, in the so-called floral meristem. A meristem is the proliferative tissue of plants. Plants grow at their tips, and the meristems are located there. Here's a representation of a typical meristem in cross-section. This is the rough equivalent of the somatic tissues of animals. The leaves grow from the crown of the meristem, as so-called leaf primordia. These are regions of tissue that develop in a slightly different way from the rest of the meristem, and they can be considered to be equivalent to the development of different somatic tissues in animals. When the time comes to make a flower, a new set of tissues develops apical to the leaf primordia. These are the floral meristems. These develop into the germinal cells of the plant. Like the leaf primordia, these also develop from the somatic tissues of the meristem. What's the separation of the germinal and somatic lines there? Let's turn the question around. Is there really so significant a difference between segregated germlines and differentiated germlines? As it turns out, not much difference, really. Let's start with a somatic chromosome. Within the chromosome, there are 4 different categories of markers and memory. First is the entire chromosome of replicable DNA, in purple. Within that chromosome are sequences of genes that are dedicated to embryonic development. Genes like those which set in motion the embryonic development of eyes. These I've coded in green. Then there are epigenetic markers that specifically influence developmental genes. Developmental markers which might accumulate to produce an eyeless phenotype, for example. I've designated these in brown. And finally, there may be epigenetic markers that accumulate during an organism's lifetime and that reflect adaptive modification of ongoing function and form within the organism. These are designated in blue. No matter whether we're talking about segregated or differentiated germlines, the germinal tissues are distinctive and set apart by a boundary of some sort. We can call this a Weismann barrier, if we like, although it might be more apt to describe it as a Weismann boundary. This is because the boundary does not block the transmission of heritable information from the soma to the germline. Rather, the boundary manages the transmission of information to the germinal chromosomes, information which significantly includes epigenetic information. For example, the entire chromosome will be replicated in the germinal tissues, irrespective of whether we're talking about segregated or differentiated germlines. Because preservation of form is an important aspect of the life history of organisms, it's likely that the developmental genes will likewise be replicated faithfully. However, not all the epigenetic markers may be transmitted across the boundary. Some may be transmitted, while others may not. If we accept that transmission of developmental epigenetic markers can be managed by the adaptive boundary, there should be no logical reason why similar management cannot also apply to transmittal of adaptive epigenetic markers. Some may be transmitted through to the germinal line, while others may not. The addition of an epigenetic perspective therefore gives us a new perspective on the nature of the boundary between somatic and germinal lines. It's not so much a barrier, as August Weismann thought. Rather, it's more of an adaptive boundary for managing the transmission of genetic and epigenetic information between the somatic and germinal tissues. The difference is crucial. The Weismann boundary is thought to be a robust defense of the modern Darwinist idea and a decisive refutation of the Lamarckian idea. Yet Weismann himself based his barrier on an erroneous concept of embryonic development. The alternative view, that the Weismann barrier is more of an adaptive boundary, opens wide the door to a coherent defense of the Lamarckian idea. Talk about irony.